myself Dr. Sunita Patra from Department of Botany, Government Nagarjun Postgraduate College of Science, Raipur. Now we are going to start with the session with primary structure of protein. So, I duly acknowledge the known and unknown sources from where I have taken the information. So, proteins they are the polymers. Polymers means there should be some monomeric units. So, what are the monomeric units of proteins? So, they are amino acids. So, polypeptides or the proteins they are made up of different type of amino acids. So, you can see the amino acids they are arranged in a planar form, chain form, they form a chain like structure. We can see the amino acids, the orange color, they are arranged by certain linkage, what we call it as peptide linkage, and you can also see a structure of amino acids. So, an amino acids consist of an amino group, a carboxylic group, R and hydrogen. Amino acids are a group of organic compounds containing two functional groups, amino group and carboxyl group. The amino group is the basic, while the carboxyl group COOH, it is the acidic in nature. There are near about 300 amino acids occur in nature, only 20 of them they occur in proteins. You can see the structure, the structure of amino acids. Each amino acids had 4 different groups attached to alpha carbon, which is the carbon atom next to COH group. These 4 groups are amino group, COOH group, hydrogen and side chain, that is R. So, for this R is different for different amino acids. Now, there are 20 different amino acids which form the proteins. So, we can categorize these amino acids into 2 categories. Plants they are capable of synthesizing all 20 amino acids, but animals they are not capable of synthesizing some of the amino acids. So, these amino acids they are known as essential amino acid, which the human beings or the animals they have to supplement through plants. So, essential amino acids they are arginine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan and valine. And non-essential amino acids, which non-essential amino acids means the human beings or animals which can synthesize these amino acids. They are alanine, aspartic acid, asparagine, cysteine, glutamic acid, glutamine, glycine, proline, serine and tyrosine. Now, these are the structures of 20 different amino acids. So, they can be categorized as polar, non-polar amino acids, aliphatic amino acids. So, amino acids they can be groups according to their characteristics. Suppose non-polar amino acids. Non-polar amino acids they are hydrophobic in nature, and these amino acids are alanine, valine, leucine, isoleucine, proline, phenylalanine, tryptophan and methionine. Then polar amino acids they are hydrophilic in nature, they are not charged, non-charged hydrophilic nature amino acids. They are glycine, serine, threonine, cysteine, tyrosine, asparagine and glycine. Now, acidic amino acids they are negatively charged, asparagine and glutamic acid. Basic group they are characterized by positively charged amino acid, lysine, arginine and histidine. So, there are 20 different amino acids total. The amino acids from where we can get these amino acids. So, suppose histidine. So, essential acids, what are the sources of different type of essential amino acids? So, you can see the histine, valine, tryptophan, threonine, the sources are given from where we can get these amino acids, essential amino acids, and we should include these essential amino acids in our diet, phenylalanine, methionine, lysine, isoleucine, so etc. So, this slide shows different sources of essential amino acids from which we can supplement this. Essential amino acid, the sources, the vegan source and the non-veg source. So, essential amino acids, why we call it as essential amino acids? Because essential amino acids can't be created by the body and have to be consumed. So, we have already said that tryptophan, threonine, phenylalanine, histidine, methionine, glycine, leucine, isoleucine and valine. So, you can get it from the sources, different sources which has been displayed through these slides. So, we have to supplement these amino acids. Now, what are peptide bonds? So, we know that proteins, they are the polymers. Polymers means the proteins, they are formed by monomers, and these monomers, they are attached to each other by a bond which is known as peptide bond. So, the carbonyl group of one amino acids, it joins with the carboxylic group of another amino acids, and it forms, the process is known as condensation, and it is known as the formation of dipeptides. Then again one amino group of one amino acid and carboxylic group of another amino acid, they combine, and thus the chain is elongated and it forms a tripeptides, and this repeated process, it forms a polymer. So, we can say this polymer as a polypeptide chain, because there is formation of many peptide bonds. There are formation of peptide bond between two different amino acids, and the process it goes on, and a polypeptide chain is formed. Now, you can see the formation of peptide bonds, amino acid 1 carboxylic group and the amino group of amino acid 2. They form, this process is known as condensation, the water molecule is discharged and the formation of peptide bonds is seen. So, what are the nature of peptide bonds? They are amide bonds. Now, these peptide bonds, what actually happens during formation of these peptide bonds? Amino acids, one amino acids, it combines with another amino acids, and there is a linear chain of amino acids which are joined together to form a polypeptide linkage, and this polypeptide linkage forms the primary structure of proteins. So, this primary structure of proteins, it is one end is N terminal, that is the amino terminal end, and the another end is C terminal end, which is known as the carboxyl terminal end. The process here is by condensation and hydrolysis, that is removal of a hydrogen, a water molecule, removal of a water molecule, and both of the amino acids, they form a dipeptide, and so on. The next, the process is repeated one by one, and one by one amino acid, they form a peptide linkage with this, and polypeptide chain is formed. Now, this is the formation of a polypeptide chain. You can see here different type of amino acids, they are linked by a peptide linkage, and you can see the amino terminal end, which is shown in blue in color, and then carboxyl terminal end, which is pink in color. So, one after the other, the amino acid, they form a peptide bond with each other, and there is a formation of a polypeptide chain. Polypeptide backbone is a repeating sequence of nitrogen, carbon, carbon, nitrogen, carbon, carbon in the polypeptide chain. The side chain or the R group is not the part of the backbone of the polypeptide chain, and you can see in the figure that the backbone of polypeptide chain is made up of nitrogen, carbon, carbon, and then and the side chain, which is the R group, they are outside and they are not the part of backbone of polypeptide chain. Now, starting, so these were the basic. Before starting with the structure of protein, we came to know that proteins, they are the polymers, and the monomeric units, they are the amino acids. Amino acids combine, or there are formation of peptide bonds between these amino acid, and they form a polypeptide chain. Now, this polypeptide chain has different level of organization. Now, the structure of proteins, it has 4 level of organization. The one is primary structure, second one is the secondary structure, third one is the tertiary structure, and fourth one is the quaternary structure. Now, you can see the structural organization of all the 4 structure. The linear sequence of amino acid, it is known as the primary structure of protein. Then secondary structure, alpha helix and beta pleated sheet, it comes under secondary structure. Then 3-dimensional conformation, it is tertiary structure of protein, and when more than one polypeptide they are involved, it is known as the quaternary structure of protein. So, these are the different structural organization of protein. Again, we can see here very clearly the primary structure of protein, that means 2 amino acids they come together to form a peptide bond in between, and it is a linear chain of amino acid. Then in the secondary structure you can see the helical structure. Helical structure is formed by, we will see in the next chapter how this secondary structure they are formed. Actually they are formed by the hydrogen bonds within the same polypeptide, and it forms a helix like structure. So, it is a secondary structure. Tertiary structure is a 3-dimensional structure, and more than one polypeptide they come together and they form a quaternary structure of protein. There are different bonds which are involved in formation of secondary, tertiary and quaternary structure of proteins. So, mainly primary structure is assembly of different type of amino acids. So, amino acid assembly is also very specific. Specific amino acid they form a linear chain. So, it is the specificity of every amino acid where it has to be placed. So, because if the sequence of amino acid is changed, the nature of protein it is changed, and it directly affects the function of the protein. So, primary structure is very basic and important and significant part of a protein, because this protein is the first organization, and after this organization secondary and tertiary and quaternary structure of protein it happens. So, it is the assembly of amino acid in the specific region in a specific linear fashion. These amino acids they are arranged, assembled. Now, then comes the secondary structure. Secondary structure is involved with the folding of these primary structures. Tertiary structure is involved with packaging. In packaging and surface to volume ratio is minimized. In quaternary structure again there is interaction between more than 2 polypeptides, 2 or more than 2 polypeptides, again minimization of surface to volume ratio. So, it is involved, it is related with interaction of more than 2 polypeptides, 2 or more than 2 polypeptides. Now, regarding structure of protein, we can go through configuration. Now, what is configuration? Configuration is geometric relation between a given set of atoms. For example, we can distinguish L amino acid and D amino acid. So, this is the different configuration L and D form. Then comes conformation. What is conformation? Conformation is spatial arrangement of atom in a protein. So, basically what actually happens, thermodynamically the most stable conformation it exists, and it is stabilized largely by weak interactions. Stability, then comes stability. Stability is the tendency to maintain a native conformation. Native conformation are stabilized by disulfide linkages and non-covalent forces. Now, what are the forces that determines the structure, such as primary structure? The forces which determines the primary structure is the peptide bond, which is a covalent bond, or we can also say it is a partial covalent bond. Then secondary, tertiary and quaternary structure, they are determined by weak forces. So, coming to primary structure of protein. The primary structure of peptides and proteins refers to the linear number and order of amino acid present in a sequence in a linear fashion. The convention of the designation of the order of amino acid is that the N terminal end, that is the end bearing the residue with a free amino group, is to the left and the number 1 amino acid, and the C terminal end, that is the end with the residue containing a free alpha carboxyl group, is to the right. So, this is the organization N terminal and C terminal end of a primary structure of protein. So, you can see in the diagram that N terminal end it starts with glycine and asparagine, and C terminal end it is asparagine. So, N terminal end means that there is an amino group, and in C terminal group there is a carboxylic group, and in between we can see a disulfide linkage between cysteine and cysteine amino acid. So, this is the primary structure of protein. It is a linear chain of amino acid containing a N terminal and a C terminal end. Now, peptide bonds. Peptide bond combines 2 amino acids and it is formed by a group of atoms from each side of the bond called the peptide group. Now, in the next diagram you can see the diagram, the portion is illustrated by a rectangle. Yes, you can see the rectangular portion, it depicts the peptide bonds, the molecules which participate in the formation of peptide bonds, it is shown in the figure. The first amino acid contributes to the peptide bonds its carbonyl carbon C1, a carbon alpha 1 and the carbonyl oxygen. The second amino acid contributes its amide nitrogen and a carbon alpha 2 and the amide hydrogen, and thus the peptide bond is formed. Now, you can see the formation of peptide bonds between two amino acids. So, carboxylic group of first amino acid and the amino group of second amino acid, there is formation, there is release of a water molecule and formation of a peptide bond, N terminal and C terminal. So, one amino acid, one end is it contains amino and the next site or the terminal end it contains a C terminal end, carboxylic group. Now, this peptide bond, it is the partial double bond character of the peptide bond. It defines the conformation of a polypeptide chain, which is very important. The peptide bond, it is partial double. So, it is the partial double bond character of the peptide bond that defines the conformation of a polypeptide chain. Now, this peptide bond, it is shorter and then a single, it is shorter than a single bond, rigid and planar. So, it is rigid. So, these are the characteristics of a peptide bond. The bond is planar and the group can take 1 to 2 more configuration. So, what are these configuration? This configuration are cis or trans. That means, the configuration, the peptide bond, it can take either the cis form configuration or it can take the trans configuration. You can see in the coming diagram. So, this is the cis configuration of two amino acids, they are joined with a peptide bond, and next one it is the trans configuration. This is the trans configuration. So, it can be either in a cis form, cis configuration or at trans configuration. Now, you can see both at a time, cis and trans peptide bonds. So, cis and trans peptide bonds, they can be well appreciated by the position of the groups. It has this peptide bonds as a double bond characters, slowly interchangeable cis and trans form. Trans form, they are heavily favored, they have heavily favored the steric interaction. Now, these are the different conformational constants we can see here in this diagram. The cis configuration of peptide group has a much greater steric hindrance between the two amino acids. That means, what is the meaning of this steric hindrance? This means that atoms get in the way of each other, and for this reason nearly all the peptide bonds in naturally occurring proteins are in trans configuration. However, cis configuration can and does naturally occur also, but maximum times it is seen that the peptide bonds, they occur trans configuration. Now, you can see the primary structure of protein. So, the primary structure of protein, it refers to the sequence of amino acids present in a polypeptide chain. So, primary structure of protein, it means by a linear sequence of amino acid present in a polypeptide chain. Amino acids are covalently linked by peptide bonds. Each component, each amino acid in this polypeptide chain, it is known as a residue, and in this primary structure we can see N terminal end and a carboxy terminal or the C terminal end. So, this is the primary structure of the protein is defined by the proteins, they are defined by unique primary sequence, because it is the primary sequence which decides the ultimate characteristics of the protein. What type of amino acids they are going to form a polypeptide chain, what are the nature of the polypeptide chain, whether they are polar, non-polar, hydrophilic, hydrophobic. So, what type of amino acids they are coming together to form a polypeptide chain, the primary structure of the protein. So, it is defined by that, and this function, it dictates the primary structure, structure defines by the primary structure, and the function is dictated by the structure, because what type of amino acid it forms a particular protein. So, accordingly the structure, the functions or they are correlated, and the functions are dictated by the structure, and structure it itself, it depends on the linear sequence of amino acids, and it is also the basis of understanding the mutation. So, primary structure is of greater importance as it defines the overall characteristics of a protein. So, this is the end of this lecture, that is the primary structure of protein. Thank you. So, coming to the secondary structure of protein. So, we were talking about the primary structure of protein. Primary structure of protein is a linear sequence of amino acids. We have seen that there are 20 different type of amino acids, and we have classified amino acids according to their characteristic feature. And this linear sequence of amino acids, specifically they are determined by the DNA, what type of code is that, and accordingly the amino acids, they come together, they form a polypeptide chain and forms the primary structure of proteins. And this primary structure of proteins, there are polypeptide bonds between each amino acid, and it forms a linear chain of amino acid, which is known as the primary structure. Now, coming to the secondary structure of protein. Now, what are the basic of secondary structure of protein? It is the polarity, it is the rigidity, and it is the planarity, which actually forms the basic structure of protein, of secondary structure of protein. Going to the secondary structure of proteins, in general proteins fall into two broad classes, structured and globular proteins or fibrous protein. In terms of structure, they are either globular proteins or the fibrous proteins. Globular proteins are compactly folded and coiled, whereas fibrous protein, they are filamentous or elongated. According to the structure, we can divide or differentiate or categorize the protein as globular, which are more compactly folded and coiled, with the filamentous or elongated structure such as fibrous protein. Now, secondary structure of proteins. What are the secondary structure of proteins? So, segment of polypeptide strands repeatedly coil or fold in a pattern which contributes to overall conformation. So, it consists of alpha helical structure, collagen helix, beta pleated sheet, turnings, non-repetitive units and super secondary structures. Then it is the three-dimensional form of the local segment of proteins, which is non-linear. So, the concept of secondary structure of protein, it was first introduced by Lang Stanford in 1952. Now, formed and stabilized by hydrogen bond, electrostatic bond and van der Waals interactions. Now, starting with alpha helical structure, secondary structure of protein. So, it is the most common conformation in secondary structure of protein. It is a spiral structure, tightly packed coiled polypeptide backbone with extending side chain. Now, this you can see the secondary structure of protein, the helical pattern of protein, how they are turned, they are coiled to form a secondary structure. Now, Pauling and Corey found that polypeptide chain with planar peptide bonds could form a right-handed helical structure by simple twist from alpha carbon to nitrogen and alpha carbon to carboxylic atom group. This helical structure is known as alpha helix, also called as classic Pauling-Corey-Basin alpha helix. You can see a right-handed helix and also a left-handed helix. The formation of alpha helix is spontaneous. It is stabilized by hydrogen bond between amide hydrogen and carbonyl oxygen group of peptide bonds. It is spaced 4 residue apart. It is very important that this alpha helical structure has a uniformity. It has a particular structure. So, it is formed, this hydrogen bonds, it is formed by, it is spaced 4 residue apart. So, every 4 residue apart we can form a hydrogen bond, which forms a alpha helix. This orientation of hydrogen bond produces a helical coiling of the peptide backbone such that the R group lies on the exterior of the helix and perpendicular to its axis. So, thus you can see here the secondary the alpha helical structure of protein, the ribbon it depicts with the alpha carbon and the side chain, and also you can see the alpha in the side diagram you can also see the mud vision which depicts the hydrogen bonds between the amino and the carboxylic group. So, in the first picture you can see the ribbon form, and in the second picture you can see the bond formation, the hydrogen bond formation with NH and CO group. Now, it is you can in this diagram you can see that the bond formation is done first and the fourth, that means first and the fourth amino acid, and this bond formation between first and fourth amino acid after each fourth amino acid, it gives a helical structure, a uniform helical structure with a pitch of 3.6. A complete turn of helix contains an average of 3.6 amino acid residues, and the distance it rises per turn is 0.54 nanometer. The R group of each amino acid residue is an alpha helix which is faced outside. So, it is a uniform structure. After fourth amino acid, the formation of a hydrogen bond, and it forms a helix, and which is the most common secondary structure of the protein. Yeah. Again you can see this how these hydrogen bonds, they are intra-chain. Just we can see the alpha helical secondary structure, it is formed by intrastructure, intra-helix, that means within the same polypeptide chain there is formation of hydrogen bonds, and it forms a helix. We can see a backbone, a spiral to which the helix pattern it turns around, and it forms a regular pattern. So, this you can see in the diagram, right handed spiral structures, and the side chain, they are outside. The pitch we have also seen, the amino acid residues per turn is 3.6, the pitch is 5.4 degree angstrom. What are the properties of alpha helix? It is a rod like structure, then tightly packed coiled polypeptide backbone core. It is a tight packed coiled polypeptide backbone core stabilized by hydrogen between NH and CO group. Side chain extends towards outside. Amino acid per turn of helix are 3.6, pitch is 5.4 degree angstroms. Hence, it gives rise as per residue 5.4 by 3.6 is 1.5 degree angstrom. This is the identity period of an alpha helix. The amino acid residue in alpha helix form have conformation with psi 60 degree and pi 45 to 50 degree. Alpha helix segments are found in many globular protein like myoglobulin and troponin C, etc. Carbonyl group of every peptide bond is in a position to form a hydrogen bond with NH group of peptide bond in the next turn of helix. Hence, it contributes to the stability of the helix. In aqueous environment, an isolated alpha helix is not stable. Coiled two identical alpha helix having repeating arrangement of non-polar side chain will twist around each other gradually and form a stable structure. So, coiled structure is found in fibrous protein. Now, this is the alpha helix structure with right hand polarity with glycine and proline amino acid. So, you can see the arrangement of hydrogen bond intra chain, intra polypeptide chain hydrogen bonds between NH and CO group. Now, these are the different type of helix. If we go for the transverse section of this helix, we can see the helix group at different organization, how compactly this helix they are arranged in different forms. You can see the compact nature of this helix. If we see the transverse section, alpha helix is seen in a alpha keratin found in skin and its appendages such as hair, nail, etc. Basic structure unit or alpha keratin is 3 right handed helical polypeptide in the left handed coil stabilized by cross linking disulfide linkages. So, alpha helix it is found in skin, hair and nails. Destabilization of alpha helix can occur as follows. The prolyl residue cannot participate in alpha helix structure, so create a sharp bend in helix. Negatively charged side chain repels one another, then steric hindrance imposed by R group, then lack of side chain in glycine. It is important. The lack of side chain on glycine allows great degree of rotation about amino acid alpha carbon. Now, collagen helix also known as type 2 helix. It is the most abundant protein in mammals. Principal structure elements of human body makes up 25 to 30% of all the body proteins. It is found in connective tissues such as tendon, cartilage, cornea of eye, etc. It contains 3 helical polypeptide, nearly 1000 residue long amino acid sequence structure is remarkably regular, nearly about 3 residues in a glycine. It contains 4 hydroxyproline and the percent composition is given, the glycine 35%, alanine 11%, proline and hydroxyproline it is 25%. Now, this is the structure of collagen, rod shaped molecule 3000 angstrom long, hydrogen bonds absent, instead stabilized by steric repulsion of pyridyl ring of proline and hydroxyproline residue. 3 strands binds each other and forms super helix, and this is known as collagen triple helix. Tight wrapping provides great tensile strength and no capacity to stretch. So, in this session we have seen primary structure of protein and secondary structure of protein. Secondary structure of protein, and now we have only discussed about the alpha helical structure. So, in the next session we are going to start the beta pleated sheets. Thank you for this session.